Pleasure to introduce Dr. Orianne Tournayre today. Orianne is a postdoctoral fellow in the Department of Biology in our lab, and has really been instrumental in a project that we have undertaken, funded by INSERT, an INSERT Frontiers project. And what we're trying to do is to develop DNA tools, metabarcoding tools, some of which Orianne will talk about today, to essentially survey all biological diversity in wetlands. In other words, that you'll be able to take a few liters of water and you'll be able to tell what birds have been there, what amphibians have been there or are there, reptiles, fish, some macrophytes and things like that. So it's a pretty exciting project, a very big undertaking, and we have lots of members of the team here. And Rojan Chen here is another postdoctoral fellow on the team. Stafford is part of this team. Alan is part of this team. Madeline is part of the team. And we're all sort of working together. But I would put Orianne at the center of this, and she's really been instrumental in putting all this together. So Orianne comes to us from Marseille, France. And that's where she contacted me from when we were looking for a postdoctoral fellow for this project. And I was very excited when I read her resume that she was interested in coming to Queen's University in January, no less. I think moving from Marseille to Kingston, Ontario, Canada in January is only for the most, those with the greatest intestinal fortitude. So Orianne did all of her degrees at Montpellier, undergraduate, master's and PhD, but she has a stunning array of experiences that many of you, even in our lab, probably are not aware of. So she did a final project on the role of chemical communication and social organization of invasive ants. That was a project, I assume, for your bachelor's degree.
Yeah.
She did an internship where she looked at ticks and fleas and the reproductive success of passerine birds. She did a gene expression study where she was looking at the biological invasion of 2 rodent species in Senegal. And then for her PhD, which I believe she's going to talk about here, she did population genetics and diet of greater horseshoe bat colonies. I will say that Orianne has really been a great pleasure to have in our lab. We just finished co-teaching a field course here. It was with Chris Eckert, who some of you might know, a botanist and evolutionary ecologist in our department, me and Orianne, and I don't think the students knew what hit them. Orianne and Chris are rock climbers, very fit. I'm less fit, but I managed to keep up. And we would put the students through their paces, getting them up at 6:30 in the morning and pushing them to 11 at night. I don't think there was a lot of partying, which was great, just exhausting. But we got the evaluations back, and the students universally said it was the best course, at least thus far, the best course that they've ever taken. And in no small reason for that was the presence of Orianne contributing to that field course, which I think was the first field course you'd ever taught. So that was wonderful. We're going to be teaching an eDNA workshop here, we hope, for professionals in July, and Orianne has actually written the entire manual, and it's been edited by Stafford and Alan, and I have yet to edit it. So really contributing in a significant way. So I am just chuffed beyond words to have Orianne in our lab. So with that, Orianne, take it away.
Thank you so much. So today I'm going to talk about my PhD work on population genetics and diet of the greater horseshoe bat in Western France. And even if I cannot present all of my results, you will have a good idea of what I've done during these 3 years. So as we all know here, there is an increase of lost biodiversity worldwide, with 1 million species at risk of extinction. And it's true for invertebrates like arthropods, but also vertebrates like mammals or amphibians. And when we think about loss of biodiversity, we always, or very often, think about charismatic vertebrate species like polar bear or tiger, but there are other taxa that are not very well known and still are threatened. For example, the bats. And the bats show a very high diversity with around 1,300 species in the world. It's 1/3 of all vertebrate species, and they have a worldwide distribution. And as we can see, they show a very high diversity in morphology, beautiful picture. but also very high diversity in habitat, in life cycle. Some will hibernate in trees, some in caves, some directly in the snow, some will migrate. And they show very diverse diets, with some eating fruits, some eating insects, and other eating blood. And those very diverse diets provide important ecosystemic services for humans. For example, pollination and seed dispersal. And if in this room, some of you like drinking tequila, you should thank the bats because they are pollinating the agave that are used to make the tequila. And they are also known to be pest control agents. One individual can eat like thousands of insects per night. So very useful for all the mosquitoes in your backyard. However, 15% of bat species are considered as threatened, and 80% have insufficient data to assess their conservation status. And obviously, like very often, the main threat is human activities with direct killing. It's especially true in tropical countries where frugivorous bats are killed because people think that they are eating all the fruits. And hunting, they are just like bushmeat. Landscape modification, pollution, disturbance and degradation of the roost, and all of that induce mortality, habitat destruction, isolation of the populations, contamination and decrease of the resources. And the consequences are loss of genetic diversity, demographic disturbance, and decrease in population size. And unfortunately, it can lead to extinction vortex, can put the species in an extinction vortex. Among the bat species, I would like to introduce you the greater horseshoe bat, Rhinolophus ferrumequinum. His name comes from the shape of his nose, a horseshoe shape. It has a distribution, like a very wide range, from Japan to Western Europe. And it has a life cycle associated to season. So it will spend the winter in natural or artificial caves. So by artificial, I mean ghost bunkers or mushroom bed. And it will just stay in torpor there the whole winter. And some mating have been observed during the season. And then in spring, they will just emerge and go to the summer site that we call maternity site, maternity roost. Actually, the female will all gather together in this maternity roost, which are often just old farm under the roof, for example. And they will gather for gestation, lactation, and the beginning of the post-lactation period where the young just start to hunt by themselves. And we don't really know where the males are. In autumn, they will just start moving again to winter sites. And most of the mating behavior have been observed during autumn. And the greater horseshoe bat is particularly vulnerable because first of its close proximity to humans. So the roost, as I said, they roost in summer in houses, in farms, but also foraging areas. It has a very short echolocation distance, just 10 meters max because of the high frequency of the echolocation. And so it needs linear landscape elements to move in the environment. And it's also, the road mortality is a big issue for this species because it's just fly at very low elevation, so just the right height to be hit by cars. It has a high longevity and low reproductive rate, so it means that the population of greater horseshoe bats have very low recovery rate. It is insectivorous, so insect decline, pollution, pesticides, climate change might impact populations. And his life cycle also is very vulnerable in winter because as other bat species, when it hibernates, we need to not disturb them because it requires a lot of energy for them to wake up. And we don't really know where they are in spring and autumn, so it's not easy to act to help the population. We don't know where they are and what they're doing and what they need, thus. In France, the greater horseshoe bat has an heterogeneous distribution. On this map, you can see all the winter data counts. So each dot represents counts, the biggest dot, the biggest counts. And what you can see here is that most of the counts are, the biggest at least, are on the western coast of France. And in 2015, a potential decline in the Poitou-Charentes region, so represented here in orange, has been observed by the Poitou-Charentes Nature NGO. And because of that, this species has been put as vulnerable on the mammal red list of this region the year after. So the Poitou-Charentes NGO and the LABEX EcoFact, which is from the University of Lyon, have initiated an interdisciplinary project to understand what is happening to this species to improve knowledge on the biology of this species. And so it's just a mix of people working on virus, on ecotoxicology, on capture, recapture, geneticists. So a lot of very different institutions and structures. And my PhD was part of this big project. I had 3 years to first delineate the populations and assess vulnerability. So just, are the colonies isolated by landscape barriers? Are they vulnerable? Can we identify decline signal in genetic data, as we observed with the count data? And then we wanted to understand if the diet could have an influence on their vulnerability. But before doing that, we had to develop a metabarcoding approach for diet analysis. And we tried to understand the influence of the life cycle and the landscape on diet diversity and composition in this region, the Poitou-Charentes region. So I'm first going to present you the results of the first chapter on population genetics. So I wanted to delineate the population and assess vulnerability. As I said before, this species has a short echolocation distance, around 10 meters, so it needs linear landscape elements to move in the environment. And that's why they love bocage, which are pasture land divided into small hedged fields interspersed with groves of trees, because they can use the trees to move and hunt. And on the contrary, open agricultural plain might act as potential barrier to the movement of this species. So the first objective was to describe genetic diversity levels, and the hypothesis was the higher isolation, the lower the diversity. Assess genetic differentiation between colonies. The colonies separated by agricultural plains will be genetically differentiated because they cannot move around. And also analyzing the genetic signature of demographic changes, but I won't present these results today. So what we did is that in 2016, we sampled 11 maternity colonies in the Poitou-Charentes region, so still there. The black maternity that we sampled. This is just a zoom of the region with the name of the cities, even if it doesn't necessarily mean something to you. But what you should look at is the colors. The green are the bocage, so, and more mid-open areas. The yellow are the agricultural plain, and the red more the vineyards. And based on preliminary results, we decided to just have a larger scale for this study. And so the next years, we sampled 12 maternity colonies in France, in Great Britain, here, in the Spanish Basque Country, north of Spain, and 1 colony in Tunisia, thanks to many collaborators, NGOs, but also academics from abroad. So what we did is that we use harp trap, so this, that we put at the entrance of the roost before dusk. So here you have some string, vertical string that you see better here. And when the bats will fly, it will slow them down and they will just drop slowly in this white U-shaped plastic containers. And so it's very nice because they can just wait all together, stay warm, and it's less stressful than being in a net. And then we just grab them and bring them to the table where we do all the measurements. So each individual that we got has been measured. Species has been identified. We collected some hair for ecotoxicological studies. We also collected parasites when they were present. So for example, ticks of this kind of diphtheria. We also collected a little piece of patagium, so the skin of the wing. And for the individuals that have the better shape, that were old enough, big enough, we also use pit tag for capture, recapture studies. And also we took some blood for virus studies. For my study, I used the patagium samples, so the wing samples, from 950 individuals, and I used 17 microsatellites, which is kind of old school now with all the NGS stuff. But just a quick reminder about the microsatellites. There are short and repetitive segments of DNA. They vary in length by the variation in the number of the repeats. So for example, repeats of the CA, and each variation is called an allele. One individual has two alleles, one from each parent. If they are the same, we talk about homozygosity, and if they're different, heterozygosity. So for example, here, allele A, 15 repeats of CA, allele B, 17 repeats of CA. So this individual is heterozygote. And thanks to that, we can study genetic diversity, genetic differentiation, and all of that kind of thing. So let's move on to the result now. First, genetic diversity. You have here the same map that I presented you before. Each dot is a colony. What we observed is a high genetic diversity, but lower at the edge. So here in Great Britain, north of France, and Tunisia. About the genetic differentiation, we use the FST, which is a common index for genetic differentiation. We observed a low FST values on the mainland, so France and Spanish Basque Country, less than 3%. And interestingly, it was higher with the colony at the very north of France that was also with a lower genetic diversity, if you remember. And as expected, the colonies separated by Cs show higher FST, so higher genetic differentiation. Then we try to assign individuals to genetic groups. I present you here the results of the structure software, but we use very different software and they all give the same pattern. So what we observed is 3 genetic groups. The first one, Great Britain, here. The second one, France and Spanish Basque Country altogether. And the third one, the Tunisian one. So just take-home messages for this chapter. Isolation by landscape barriers. While we observed one stable, I didn't present the result for that, but population was stable. And large in France and Spanish Basque Country. So what we thought was not true. Agricole plain and large river don't seem to have an effect on the movement. At least we could not detect it in the genetic data. Whereas seas and or mountains could be potential geographic barriers, as it has been shown in another Rhinolophus species, the lesser horseshoe bat. Are they vulnerable? Well, we observed a lower genetic diversity and a higher genetic differentiation in north of France and Great Britain, so they potentially could be more vulnerable. But with our sampling effort, we don't know if it's a local effect or an edge effect, because it was at the edge of the range of this species. So now I'm gonna talk about diet. Is there an influence of the diet on roost colonies' vulnerability? But first, why the diet is important in bat conservation? Well, first, because insectivorous bats have high energetic needs to fly, for echolocation, to hibernate, or even for breeding. As you can see on this picture, the female is carrying the baby. The baby is like 1/3 of his size. It's attached to false nipple, so very heavy, and we can imagine easily that it's not easy to fly with that attached. So that's why they need a high quality of prey, for example, rich in fat acids or calcium for hibernation and lactation, but also a high quantity of prey. It has been shown that some individuals need it's between 20 and 70% of their weight in insects every night. So just to give you an idea of what it represents, for a human of 80 kilograms, it's up to 60 kilograms of food per day. And we do know, thanks to all the studies in the literature, that there is an influence of the quality and quantity of prey on fitness. At least it has been shown on other insectivorous species. This study was on birds. And the issue is that because of habitat destruction and the massive use of pesticide and anti-helminthics, so just drugs for cattle against worms, there is a decrease of biodiversity and biomass of arthropods, but also a contamination of prey, the arthropod, and bats through bioaccumulation. And so does it have an impact on the populations? But how to study bat diets? Obviously, we cannot determine the prey just by looking at the bats flying and hunting, like insects are too tiny. So what we do is that we use the fecal samples, and there are two ways to get the fecal sample. The first one is capturing the bat. So here you can see the fecal sample, tiny in the tube. And it's great because it gives us all the individual information: bat species, the sex, the age, body condition, anything. But the issue is that it's very invasive, like we have to capture the bat for that and handle them. And also we got a low number of samples because they don't always have poop. So the other way of getting fecal sample is just collecting the samples beneath the colony. So what we do is that we use disposable plates, as you can see here, and all the black dots are fecal samples. And when all the bats are gone to hunt at night, when the roost is empty, we just go and collect the fecal sample using disposable tweezers to not have contamination between the samples. And it's great because that way we have like hundreds of samples. It's not invasive, at least it's not disturbing the bat as much as the capture. But the issue is we don't have any individual information unless we do genetic studies to know the sex and that kind of thing. So now we know how to get a fecal sample, but how to identify the prey in a fecal sample. The traditional approach was to just use the microscopic identification of the hard parts of the insect. So on this picture from Claire et al., you can see the legs of a Lepidoptera, so we know that this individual ate a Lepidoptera. The issue is we cannot know all the soft-bodied insects that have been eaten. It has a low taxonomic resolution, like how to get to the species level with just a broken leg. Sometimes it's possible, but most of the time it's not. It's very time-consuming, and taxonomic expertise is critical. So since 10, 15 years now, it has been developed a molecular identification of the mixed DNA in the samples. So we extract the DNA from the samples. We amplify a short fragment of this DNA. I say short because DNA is degraded. So if we want to amplify a too long fragment, it won't work. So short fragment amplification, we sequence it. Then we use bioinformatics and filtering to get rid of all the contamination, sequencing errors. And we compare the clean sequences to the sequences available in reference database. And we can have the list of species of what this individual has eaten. However, most of the time, and it's the case for the greater horseshoe bat, there are mixed colonies. So different bat species will share the same roost. And also other insectary roosts, like the swallows, barn swallows, can share the roost. So we need to identify also the predator species, not just the insects, but also the bat species, to make sure that we attribute the prey to the right predator. And that's what we tried to do during my PhD, to develop an approach to amplify both. And to do so, we used in silico analysis, so just using all the sequences available in public reference database. We use mock communities, so DNA from insects and bats that we identified and put artificially together just to see if we can identify all of them and at the species level. And we use guano samples from 16 bat species that we captured in 2016, but also guano that we collected non-invasively, so just beneath the colonies. And it worked well. So great, we can now identify both hard and soft-bodied insects. We have a high taxonomic resolution to the species level most of the time. We can identify the species of both the prey and the predator. The issue is that it's quite expensive, and also it requires specific equipment, a clean environment, lab. So it's not accessible to everyone unless collaborations are made, or fee-for-service lab can also provide this kind of analysis. So we know how to get the sample, how to identify the prey, and what do we already know on the greater horseshoe bat diet? Well, based on studies made in Great Britain using microscopic identification, we know that this bat species feed mainly on Lepidoptera, Coleoptera, and Diptera, and that there is a variation in the proportion of this taxa, depending on the abundance of Lepidoptera in the environment. And we also know that the species can make a prey selection decision based on prey size and density, and so potentially can fit with the optimal foraging theory, which is the individual will spend energy to capture the prey, to eat the prey, and we gain energy by eating the prey. And so there is a trade-off to maximize the energy gain. Opportunist species or individual will minimize energy expense, whereas the selective individuals will maximize energy gain. So instead of just getting everything, they will focus on just some prey that provide them a lot of energy. And we had two hypotheses. The first one was temporal. We know that in June and July, there is a gestation and lactation, a peak of energetic needs, but also energetic constraints. Remember this baby that the mother is carrying, or during lactation, when the females have to come in and out of the roost several times per night to feed the babies. So this might lead to a more opportunistic diet, which will be translated by high diet diversity and a strong phenology effect on diet composition. And we do know that the prey availability vary according to the landscape. So there would be a higher variation in diet composition between roost colonies located in different landscapes. So what we did is that in 2018, we sampled in 7 maternity roosts still in the Poitou-Charentes region. Each bat here shows you the colonies that we sampled. We used a non-invasive way of collecting samples, so using the disposable plates beneath the colonies. And we sampled once in June during gestation, once in July during lactation, and once in August when the young start to feed by themselves, and everyone is just preparing the winter, getting fat reserve, and preparing for mating. And in total, we got 1,995 samples. But as I said, the greater horseshoe bats often share the same roost with other species, and mainly the Geoffroy's bat. So it was very useful to know the predator, to have the identification of the predator. We kept only the sample that showed an amplification of both the bat and the prey DNA. We discarded all the samples that had DNA from other insectivorous species. So we got some swallows, other bird, rodents, other bat species. We just discarded all of those ones. We just kept the 100% greater horseshoe bat samples. We discarded samples that showed contamination or inconsistent detection. And so in total, we had 1,033 greater horseshoe bat samples from 6 colonies. Here, I would like to show you how we try to define the landscape for each roost. So you have here a PCA. Each bat is a colony, and the variable landscape variable are represented here with the arrows. What we observed is that the first axis, the first dimension that explains 50% of the variance, show the gradient from forest, so close coniferous wood forest, close mixed forest, to mid-open habitat, so more bocage with meadows and hedgerows. And the second axis, dimension of the PCA, that explain around 30% of the variance, showed that this colony was characterized by vineyard, forest and vineyard. We observed between 1 and 90 preys per sample with a median of 4. We described 17 orders and 519 species. 57% of them were Lepidoptera, 23% were Diptera, and 13% were Coleoptera. So here is the occurrence in the world diet, I didn't say, and all the orders that we detected. And thanks to the Artemis database, which contains 2,000-ish pest species, at least in 2019 when we used it, we could describe in the diet of these colonies in the Poitou-Charentes region 133 pest species. Most of the prey in the diet, in the data set, are rare, a median of 2 occurrences. So just to give you more detail, 300 taxa are just 1 occurrence in the whole data set, so the 1,000 of samples. And 103 taxa had just 2 occurrences, so very low, very rare prey. And the 15 most common species had between 50 and 106 occurrences. On this graph, I'm going to talk about diet richness now. You can see the Shannon diversity on the y-axis. For this one, this graph, you have the first axis of the PCA, so the gradient from forest to bocage landscape. And on this one, just a month of sampling. And what we observed is an effect of the month and the landscape on diet richness. Diet richness increase from forest to bocage landscape, and there is a peak of diversity in July. We also observed for the diet composition that where prey exacerbate the dissimilarity between roosts. So what I didn't say is that for all the rest of the presentation, the sandwich just represents the 15 most common species. The little candy, all the very rare species. So when you see a sandwich and a candy, it's the whole data set. And when you see just a sandwich, I'm speaking about just the most common species. So we observe that the rare prey exacerbate dissimilarity between the roosts, and that there is a high overlap between the different months of sampling for the most common species. As you can see here, each dot, triangle, square is a sample from the different colonies here, and each color is a month of sampling, and you can see that there is a big overlap, but still a gradient from June to August. June and August share less prey species than June and July together and July and August together. If we talk about now just about the common species, in the 4 colonies that are in the bocage landscape, we observed an homogeneity of the pattern in the diet composition. You have here the percentage, the proportion of each of the 15 species. The pink are the Diptera, the more yellow are Coleoptera, and the blue are the Lepidoptera for each month of sampling. And what we observe is a peak of Diptera, so the red arrows, in each of these colonies, and a decrease of Lepidoptera in July. We observed a phenology effect. For example, this Lepidoptera species, Agrotis bigrama, only flies in August and September, and indeed we find it just in August. But also potentially some selectivity. This beetle is occurring the whole summer, June, July, August, and we don't detect it all the time. For example, here we detect it just in June and August, as it is the case in those two. In this one, we detect it in during the 3 months, but much less in August. For the 2 other colonies, the one characterized by forest and the one characterized by forest and vineyards, the pattern is heterogeneous. They don't show the same thing. The first colony, this one, show a lot of Coleoptera in yellow and Lepidoptera in blue, whereas the other one show a similar pattern than in the bocage. So we did find a high taxonomic diversity, much more diverse than ever observed using microscopic identification, with a much better resolution to the species level. We observed that a majority a lot of occurrences. Potentially, this very rare prey can complement a core diet, the very common species. And we observed a strong effect of the month of sampling in the landscape on diet richness and composition. Potentially an effect of both phenology and selectivity in the bocage landscape. The individual could be more opportunist in July because of the high cost of lactation, so increase in diet diversity, and more selective in August because they don't have the same energetic constraints, so they can be more picky. And we don't really know about the colonies situated in forests because we had just 2 colonies. They showed very different pattern. We would need more colonies to be able to say something there. What we do need to test those hypotheses of phenology and selectivity is the prey availability and profitability. Without that, we can just have assumptions. And there is an importance of the landscape in the prey availability. For example, the light might attract insects, but so species that don't, bat species that don't like the light won't come here, won't have access to these resources. But also the importance of temporal replicates, because seasonal pattern can change from one year to another year. It has been shown in other bat species. And also, depending on the temperature, the insects might emerge later, earlier. So we need to have more replicates in time to be able to have the biggest picture. So to conclude on my work for this PhD, the question was, does the greater horseshoe bat show vulnerability signals? We didn't observe that in Poitou-Charentes region, not at the population scale. So France and these colonies, 6 colonies here, no decline signal, high genetic diversity, high gene flow, and actually the NGO that made the first observation of a decline of 30% based on count data, redid the analysis with a more robust model and actually found no signal of decline in the data. However, this work suggests large movements, but what are the mechanisms? Is it a seasonal dispersal, natal dispersal? When and where are the individuals when they are mating? Because we just observed the individual when they are gathering for the winter. And so to answer those questions, the capture mark recapture study is ongoing, and they just get a lot of data over the years to understand what is happening, where are they moving, and so on. And even if everything seems fine at the population scale, we found a lower genetic diversity and higher genetic differentiation here at the very north of France, which is at the northern edge of the population. And so, as I said earlier, we need more samples to know if it's a local pattern or if it's an edge pattern. And it's especially important to study because during the 20th century, in Belgium, in Luxembourg, in Great Britain, there was a huge decline of the populations. And so the question of the diet is interesting if there is this edge effect, because it has been shown in a bird species that, I don't know if you can see where it's maybe too tiny, but you have here the diet breadth according to the distance to the edge. So the more on the left you are, the closer to the edge you are. And here you have the breeding success and the same distance to the edge. And what we can see is that the diet breadth increase at the edge, but the breeding success decrease. So you might hear it's a more diverse, have a more diverse diet. It doesn't mean that it's good. Like it doesn't mean that it provides all the things you need to have a good reproductive success, a good breeding success. So with all of that, I would like to thank my PhD supervisor, Nathalie Charbonnel and Dominique Poncher, all the people that have been involved in the NGO, in the different academic institutions I worked with, and obviously all the field volunteers and collaborators. Thank you.